Imagine that you are conducting an investigation in which blood cells are placed in 3 solutions, each of which contains a different amount of salt as its solute. How does the flow of water, or osmosis, affect a blood cell which has been placed in different types of solutions? First, we place a blood cell in a hypotonic solution. Hypotonic means there is a higher concentration of salt inside the cell than in the solution. Water is an equalizer, so it is going to want to dilute the salt inside the cell. The water moves across the membrane via osmosis and begins to fill the cell. If the cell becomes too full of water, it will burst or lyse, just like an overfilled balloon, and kill the cell. It is possible for organisms to die of overhydration. Next, a blood cell is placed in a solution in which the amount of salt is equivalent to the amount of salt in the cytoplasm of the cell. We call such a solution isotonic. Water loves equality. It will flow towards the area which has the lowest proportion of water to salt. This movement along a concentration gradient is called osmosis. In an isotonic solution, the water-to-salt ratio is already equal, so water flows into and out of the cell, and the cell remains healthy. We all know that drinking excessive salt water can be deadly, but why? Seawater is a hypertonic solution. In a hypertonic solution, there is a higher concentration of salt outside the cell. As a result, water moves out of the cell via osmosis. As the water moves out, the cell begins to shrivel. Remember that the liquid environment of the cell is analogous to our air. If our air is depleted, we will not survive. Likewise, if the cell's water level is reduced, it will die. Let's review. Water moves from areas of low solute concentration to areas of high solute concentration. In other words, water will follow the salt. In a hypertonic solution, water follows the salt into the cell, causing the cell to lyse or burst. In an isotonic solution, the salt concentration inside the cell is equal to the salt concentration outside the cell, so the water does not have a net flow in either direction. In the hypertonic solution, the salt concentration is higher outside the cell. The water moves from the cell to the solution, causing the cell to shrivel.